Here we go with the video. You saw the title. A 2014 mitochondrial DNA test by Collective Evolution, Ancient Origins, and Facebook prove the 1974 Paracas skulls are non-human. But then again, the same sort of test would prove that roast beef was non-human. And also any other skull from the time, but let's continue. Claim they're 3,000 years old. Doctors that someone had spoken to said that you can alter the shape of a skull, but you can't increase the size of a skull, and it's genetically predetermined to have a certain volume. Neither of those is accurate at all, by the way. But let's continue. Later tests contradicted earlier results because it was a BS story in 2014, but we must repeat the chant, because what happens is you recopy the same fake story, and that's all it takes. Just if you repeat a lie often enough, it becomes the truth, right? And here we go. Initial DNA analysis of them has revealed that they may not have come from humans, but from a completely new species. This would require not that you find that they're just not human, or that you can't prove they're human. You'd have to prove that they were literally something else identifiable. And it would also require that you don't hide who the geneticist was. Whatever, and by the way, I don't know if you know this, but these skulls are not uncommon. They're around. I'll get to why in a minute, but other people tested other skulls that look exactly the same, but these are special skulls. Right. Which ones? We're not telling. Anyway, let's continue. Whatever the sample labeled 3A has come from, that means the person didn't know, let's say this is a double-blind test, it had mtDNA with mutations unknown in any human. Remember the word mutation. Okay? Primate or animal known so far. So this is the problem. Any animal, any primate, any human. How many chromosomes? Okay. How many base pairs was it? You know it has more DNA in it than you? Some types of shellfish and a lot of bugs and some viruses. Yeah, the number of DNA strand, the number of base pairs, the length of it, complexity, if it has multiple chromosomes, if it has duplicates. Where's the rest of the data? It's just not a human. That's nice. What about the other ridiculously large number of creatures on the planet? And what about you saying it's 3,000 years old? Next, the data are, that's technically okay grammar, the data are very sketchy. And a lot of sequencing still needs to be done. Wait a minute. You've concluded that this can't be a human, but you haven't done all the sequencing? If you sequence a chimpanzee, unless you start it from the right spot, it will match a human for the majority of it. Because it literally does. Because it does. I'm sorry, but what the hell are you talking about? Okay, let's go on. We need to recover the complete mitochondria DNA sequence. Then how do you know that it isn't a human's mitochondria DNA? Because mitochondria DNA is a bit different. A few fragments I was able to sequence. Oh, you only sampled a few fragments from sample 3A. Indicate that if these mutations will hold, we are dealing with a new human-like creature. But if you only have a partial sequence and it's mutated, how do you know it was human? If you said it came from something called sample 3A, but you weren't told what it was, that's bullshit. If you were not told what the sequence came from, you'd say, I can't identify what it's from because I have fragments of only the mitochondrial DNA. You wouldn't come to the conclusion whether or not it's human or not. You'd say, it's probably, I'm sorry, but this is what happens. If someone hands you a muffler and asks you what car it comes from, but it's only part of a muffler, and doesn't tell you what car it comes from, but they tell you, we don't know what kind of animal it came from, the first thing out of your mouth is, it's not an animal, it's a muffler from a Buick. This is not the answer you would get from a geneticist. It's not a geneticist because they didn't, unless there was an explicit request, is this a human? The answer should be, if you only have fragmented DNA, I can't tell, there's not enough data. This is like somebody handing you, I'm not kidding, handing you a data file they got off the net of random characters, and you're supposed to figure out what kind of file it's from. Unless it has a really good earmark, the only thing you can determine is that it's not a text file, and even that won't work. It is actually the same thing. If you take a 4 gigabyte thumb drive and give me, at this point, 100 bytes of data out of it, if I get lucky, I'll guess what it is, and it might actually be accurate. But it could be a text file. It could be the text file that's embedded in the DVD for the subtitles. It could be anything. This is bullcrap. Anyway, let's go on. Very distant from Homo sapiens. If you don't have enough DNA to distinguish what the hell it is, then you don't know if it's related to Homo sapiens, Neanderthals, or whatever. I'm not sure it will even fit into known evolutionary tree. How would you know that if you don't have enough? Look up how long the DNA sequence is for mitochondrial DNA and what other creatures have similar lengths. Let's go on. And what cell did he get it from? I'm not kidding. It's supposed to be from a skull. What part? Was he told what part it was? Was he told it was supposed to be a human? And again, if you have partial DNA fragment, you wouldn't assume it's a human. You would just assume it's contamination from another DNA source. Breeding within their small population, they may have degenerated due to inbreeding. That would explain buried children. Wait a minute, all he got was a DNA sequence and he's coming up with this? Whatever 3A came from. Okay, this is not a double-blind check, and this is not a geneticist. Can we stop this? Please. They were either low or not viable. Low or not viable? I didn't read that part. Anyway, let's actually get some information here. For context. Context. Anyway, here we go. The Nazca culture came from and developed directly out of an earlier culture. The Paracas, it could actually be Paracas, Andean culture in the Andes Mountains that existed until 100 BC. Okay. How early? Have to look it up. Is it really 3,000 years old? Okay. Let's assume on purpose that it's not 3,000 years old. Let's assume that it's the minimum number of centuries old. Let's assume it's 2,100 years old. 2,100 years old. Let's assume it's the last of their culture. There's a reason. DNA without enzymatic repair, living cells, has a limited chemical stability because it's a sequence of amino acids. Adenine, guanine, thymine, cytosine, uracil. I can't pronounce the last one. Uracil. It's Gattaca and U. U is used for messenger mRNA for transcribing it. Go look up the stuff. Anyway, here we go. A large number of post-mortem mutations increase with time after a cell has died. After a cell has stopped functioning, without the cell actively correcting DNA changes, it might go through 1 or 2 cell division attempts and fail, and you'll have broken DNA chains. Also, the DNA in your cells isn't just used to be DNA, it's used to copy data from. It's also decoded and re-encoded for making proteins. As long as the cell is kind of functional after something dies, it'll keep doing that until it comes to a stop halfway through or something. And this means that the cell is chemically active at death. Cell activity lasts for however long it does, until the cells starve for oxygen or whatever. And then the bacteria in the digestive system begins to work, and the surrounding environment if something was buried. See how this is starting to sound a little hokey? But let's continue. Warmer tropical climate degrades DNA quickly. We also call that decay, rotting, whatever. Not to mention things eating it, including bacteria and microbes and ants. Miscoding of cytosine to thymine and guanine to adenine, that's C to A and G to A, accounts for the majority of errors you find. If you're not aware of it, that's a pretty big error. Those are the paired upsets. So you get broken DNA base pairs. They're not decodable after a period of time, but you can determine what they probably were by kind of inferring, okay, Let's assume they flipped. I mean, this is the same thing you would do with a database. You can actually do this kind of. But let's continue. DNA can theoretically survive, meaning being detectable as being something, and if it's chromosomes, at least you can count the chromosomes and figure out the length and figure out what it probably was. You're not going to get a detail out of the DNA chain, but you can figure out whether it was a human or not by the number of chromosomes. There's no mention of that. Why not? Well, it's mitochondrial DNA. Okay. How much code's in a mitochondrial DNA? You have to look that up. Let's continue. But that's theoretical. For about 1 million years. So that means you can find that it was DNA rather than just a patch of something else if the cell, the whole thing, or at least part of it, didn't get screwed with. And this is mostly plants. This is mostly bugs. Things stuck in amber, literally. Not a dead body that's the flesh it's been defleshed and the bones have been sitting out for a long time. No, not even dinosaurs. They're more than 1 million years old, by the way. But let's continue. At 6.8 million years, due to something I'm going to discuss with you in a minute, all bonds would be broken. Why? I'll tell you. Early 1990s claims for real recovered million-year-old fossil DNA were from contaminants, because eventually they were able to sequence them, because we eventually got to where we could do that and found out it was a modern critter. And it didn't match the bones, so it's contamination from like a housefly or something. Or bacteria from, something digesting it 1 million years ago could have been there. Who knows? DNA from a bone, which is what we're talking about, these are skulls, has a half-life of 521 years. That means after 521 years, not 512, not the binary number, but pretty dang close, so I can do this almost in my head, half of the DNA chain base pairs are broken or flipped. The bit flip factor is a big deal, but they'll break, which means the DNA chains will break into smaller pieces. And it isn't at specific points. The whole thing starts to break apart in hundreds of locations. Individual base paired groupings break loose. So it stops being a DNA chain or a chromosome, and it turns into individual fragments of basically proteins. Amino acid protein chains. Now you're diagnosing protein sets. And the smaller it gets, the more it looks like a virus. But let's continue. DNA from bone has that half-life. What does that mean after 2100 years? That's 4 half-lives, approximately. I'm going to do 512, 128, 256. You get the idea. So it's 512, 1K, 2K. That's 4 max. So let's say 3 or 4 half-lives. Let's just do that. It's like 5. Yeah. You're going to lose anywhere from, well, up to 94% of the DNA is lost at that point. That sounds like what we got here, doesn't it? It's not an inhuman, it's data loss. And anybody who's a geneticist would know that after seeing not enough DNA to identify it, you shouldn't decide that it was never human. You wouldn't even know if it was a plant. The Peruvian samples, there are no statistically significant difference between artificially deformed skulls, which is a tradition in the area and on other places on this planet, and normal skulls. 400 BC is the earliest written record by Hippocrates, or Hippocrates. Hippocrates. Excellent! Anyway, let's just get her down here. These were DNA fragments, and we're supposed to assume a geneticist wouldn't know a DNA fragment, can't really tell you what it is if you don't have the rest of the data. Whatever this was, it wasn't a human. Why would you assume just mitochondrial DNA that you found somewhere is to that? It's just an animal cell. Mitochondrial DNA is in animal cells. That's an animal cell. You can look up all the different numbers of DNA. What else was given? Nothing. This story was touted by a bunch of people who didn't care if they were called out for being ignorant. Please don't repost the story. Thanks for watching. Have a good day. Good luck with that. And yeah, that's the origin story for another one. Bye.